Hello and welcome everyone to this podcast. In this podcast we're going to focus on the operon. Several decades ago there were two French scientists. They were called Jacob and Monod. And they were interested in how bacteria change their regulation in order to use glucose or lactose or other sugars. They quickly learned that if you give E. coli glucose, that they would grow and use that glucose. They also learned if they gave it lactose, it would also grow. They also learned that if they gave it glucose plus lactose, it would grow, but it would use glucose first. Now this seemed like a great opportunity to understand a basic fundamental pathway. E. coli was capable of metabolizing both of these sugars, but when both were present, it shut down lactose metabolism so it could metabolize glucose. And then when all the glucose was used up, it would start metabolizing lactose. So let's draw a cell here, an E. coli cell, and we have lactose on the outside of the cell. And to get it into the cell, there is a protein that forms a channel here, which allows lactose to move into the cell. Galactose could then be broken down into galactose and glucose. One thing I should have mentioned is that lactose is a disaccharide. Meaning it contains two sugars attached to each other. Galactose and glucose are both monosaccharides. To use one of these more complex sugars, it almost always has to be broken down into glucose or galactose or some other monosaccharide. It is broken down into glucose and galactose using an enzyme called beta-galactosidase. And this enzyme functions right here by converting lactose into galactose and glucose. So how does it regulate these genes? E. coli and other bacteria packaged their genes into a common operon. So let's talk about an operon first. So let's draw an operon. The operon, like most genes, is going to have a promoter here, and something that's a little unique to bacterial genes or other organisms that use an operon is that they have an operator. Let me write that out. Operator. That is positioned between the promoter and the genes. Now the thing that makes this system work, and makes it work really nicely, is that there will be multiple genes right next to each other. Now, for now, let's just call these A, B, and C. When a RNA polymerase binds here and makes messenger RNA, what it actually makes from these genes is a single messenger RNA. And on this messenger RNA, right next to each other, you have gene A, or the mRNA from gene A, and then the mRNA from gene B, and the mRNA from gene C. Now what happens is the ribosomes bind here, and they'll bind here, they'll bind here, to begin making proteins from each of these genes. So by transcribing, an advantage to this is that by transcribing one long messenger RNA, you're able to make this one messenger RNA and able to make 3 genes at the same time, or more than 3 genes, just depending on how big the operon is. These operons, or the genes within the operon, very importantly, tend to have, or I should say tend to function in a common pathway. So we're going to talk about the lac operon in a moment, so we might as well just talk about that here. With the lac operon, it encodes 3 genes that makes 3 proteins, and each of these proteins is important in metabolizing lactose. Other operons might be turned on and they're important in metabolizing arabinose, another sugar. So it's a way to kind of cluster the genes that need to function together in one spot. And now the convenient thing about this now is when you want to start metabolizing something like lactose, all you have to do is turn on one gene, and now it's going to make all 3 of these proteins. If these 3 genes were regulated by their own individual promoters, such as something like this, with your promoter here, and say this was gene A, then you had another gene with a promoter, and gene B, and then still another gene with a promoter, and gene C. In order to metabolize lactose in this description here, the cell would have to turn on each of these genes at the same time. It's much more efficient if you can just turn on one gene, one promoter, and activate all these genes at the same time. So while we're here, we might as well define 2 terms, and they are polycistronic and monocistronic. Polycistronic is the case we see up here with our bacterial cells, where they will make a single messenger RNA that's able to code for multiple proteins. Poly. One mRNA, multiple proteins. Eukaryotes usually have monocistronic messenger RNAs, much like we see up here. They code for a single protein, mono. The last thing I want to say about this operon is that there's a regulatory protein down here. It could be an activator or it could be an inducer, but it is regulated by its own promoter. In this case, it's going to make a monocistronic RNA. So this regulatory protein gene here will make a protein, again either an inhibitor or an activator. And this inhibitor or activator will now act upon this gene. It's important that it's not part of the operon, because if it was a part of the operon, how would you ever turn it on? How would you ever get the inhibitor or activator? So it's important that it's under a separate control, under a different regular. Next, I'd like to take this operon and make it more specific to lactose, or the lac operon. Let's draw our gene. We're going to have our regulatory protein down here, our regulatory gene, I should say. Then we're going to have our promoter here, followed by an operator, followed by 3 genes. That we're going to call gene Z, gene Y, and then finally gene A. And just like we saw in the last slide, when this gene is activated, it's going to make a polycistronic message so that it can make all of these proteins from the single messenger RNA. This regulatory gene in this case codes for a protein called LAC-I. It's a repressor. Now what this repressor does is binds to this operator. Remember, this system is usually turned off because bacteria are going to prefer glucose, so it's usually off. So with that in mind, we should call it what it is. This is an inducible system. It's usually off, but we're going to find a way that we can turn it on. So the LAC inhibitor binds to this operator. And when this LAC inhibitor is bound to the operator, remember that means our RNA polymerase can't move past this operator. The operator can't move past this repressor that's bound to the operator. The repressor serves as a roadblock, keeping the RNA polymerase from transcribing these genes. So it's usually off, and this is when there's no lactose in the cell. So no lactose. So with no lactose, repressor is bound to the operator, no transcription of the genes Z, Y, A. Now, what happens when lactose enters the cell? So now we have lactose present. Well, what lactose does, it serves as an inducer. It will bind to this repressor, and when it binds to the repressor, let's redraw the gene down here to show you what it looks like once lactose enters the cell. Okay, so lactose enters the cell, it binds to this repressor. When it binds to the repressor, our $1 million word happens, that allosteric change occurs to the repressor. The repressor now falls off with the lactose inducer bound to it. Once it falls off, RNA polymerase can now bind to the promoter and begin to transcribe these genes. Okay, so let's go over this one more time, just real brief here. We have our lac operon. It is an inducible system using a repressor. The repressor is bound to the operator when there's no lactose present. Remember, we want this to happen because there's no reason to turn these genes on if there's no lactose. E. coli are incredibly efficient cells. There's no reason to turn on genes that you're not going to use. It's costly. It costs a lot of cellular energy to do that. So it keeps it off. Now, when lactose enters the cell, shown here, it will bind to the RNA polymerase. Once it binds to the RNA polymerase, it falls off due to an allosteric change. Now, without the repressor bound, the RNA polymerase can now transcribe these genes, Z, Y, and A. And now Z, Y, and A can make the specific proteins. I realize I didn't say what these coded for. I kind of alluded to it, but I should tell you specifically. Gene Z here makes beta galactosidase. Remember, that's the gene that's going to break lactose down to glucose and galactose, usable sugars. Gene Y is a permease. Remember that protein that brings lactose into the cell. Gene A here is a transacetylase. And we're not going to talk about this. Transacetylase. Focus on beta-galactosidase and permease. Okay, I'm going to erase this real quick here because I want to pose a problem that the cell encounters. And it's involving this gene right here, permease. Remember I said you have your E. coli here, and the way that E. coli gets into the cell is using this permease. That seems like a great system. Now the problem comes though when we look at the genes. I'm going to just briefly write the operon down here. We have our Z, Y, and A genes controlled by this promoter and this operator. And remember, this repressor is bound to the operator. And the only way to get this repressor off, to make beta-galactosidase and to make permease, is for lactose to bind here and remove it. The question is, how do we ever get lactose into the cell to pull this repressor off if we never transcribe these genes? Remember, this permease is made by this operon. And so permease isn't usually in the cell membrane. So there's got to be a way to get lactose into the cell so that we can fully pull this repressor off and turn these genes on. But it seems like it's impossible to do that because the key to it is right here. I'm not going to answer that right now. We'll talk about that in class. How does the cell get over this problem? Okay, I'm going to break this particular objective up into 2 podcasts because I think this makes a nice cohesive story here, just talking about lactose metabolism. Then in the next podcast, what I want to talk about is how we use this operon and looked at lac operon mutants to further understand lactose metabolism, or just in general, these pathways that help the cells survive. So let's have a quick summary here. We defined a few things here. We defined the operon, in general what it is, and how that was able to make a single messenger RNA that then could be used to make multiple proteins, and how that the advantage to that is that the system only has to turn on one promoter in order to turn on a whole series of genes necessary for a single pathway. We talked in great detail about a specific operon called the Lac operon. And we talked about how the Lac repressor bound to the operator to prevent transcription, and then how lactose pulled that off to make those genes. We closed with a question then, just a moment ago, about how we can get lactose into the cell in order to turn the gene on eventually. In the next podcast, we're going to take this Lac operon and pull it apart a little bit more by looking at mutants. That's all I have for now. If you have any questions, please let me know. I'll see you in class. Bye.